Hello, everyone, and welcome to today's webinar, Leveraging Single-Cell Copy Number to Resolve Tumor Clonality. I am Michelle Ashton of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by 10x Genomics. To learn more about our sponsor, please visit www.10xgenomics.com. Let's get started. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. To do so, simply type them into the Ask a Question box and click Send. If you have trouble seeing or hearing the presentation, click on the Support tab found at the top right of the presentation window, or report your problem by clicking on the Ask a Question box located on the far left of your screen. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located at the top right of the presentation window and follow the process to obtain your credits. I now present today's speaker, Dr. David Wesley Craig, Professor of Translational Genomics and Co-Director, Institute of Translational Genomics, Keck School of Medicine of USC. For a complete biography of our speaker, please visit the Biography tab at the top of your screen. Dr. Craig, you may now begin your presentation. Hello, everyone, and welcome to today's webinar, Leveraging Single-Cell Copy Number to Resolve Tumor Clonality. I am Michelle Ashton of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by 10x Genomics. To learn more about our sponsor, please visit www.10xgenomics.com. Let's get started. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. To do so, simply type them into the Ask a Question box and click Send. If you have trouble seeing or hearing the presentation, click on the Support tab found at the top right of the presentation window, or report your problem by clicking on the Ask a Question box located on the far left of your screen. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located at the top right of the presentation window and follow the process to obtain your credits. I now present today's speaker, Dr. David Wesley Craig, Professor of Translational Genomics and Co-Director, Institute of Translational Genomics, Keck School of Medicine of USC. For a complete biography of our speaker, please visit the Biography tab at the top of your screen. Dr. Craig, you may now begin your presentation.
Okay, I'm going to go ahead and get started. Hello, my name is David Craig, and I am from the University of Southern California within the Department and Institute of Translational Genomics, and I'm really excited to talk about some of the work we've been doing looking at how we can leverage single-cell copy number to resolve tumor clonality and better understand heterogeneity. I kind of want to start with this slide. It gives you an overview of where I come from, and I study kind of a broad swath of different diseases, such as inherited disease, such as cancer, as well as diseases of aging. But underneath all of it is really technology, and technology is enabling. And it's these measurements that have really given us a lot of insight. And that's really where our expertise is on, where we work a lot on the informatics, the data sharing, and the technology. Now, the past decade has seen just a revolution of capabilities, and it's all come down to kind of one principle that matters, and it's this idea of we can better resolve heterogeneity if we can measure things on their atomic, on their elemental, on their singular kind of value. And that's what brings me here, is the idea we've spent a lot of time looking at sequencing, such as this, and now we're able to look at sequencing in the context of a single cell. And that, I believe, is going to be enabling. And so this talk, we're going to talk and look into understanding heterogeneity. And so I think there's this inherent understanding that cancer in itself is usually not just one thing. There's a lot going on. There's actually a lot of different models of cancer heterogeneity. There's heterogeneity and intra-tumor heterogeneity. And these models have kind of this idea that there's a evolution that's ongoing. And this evolution is one that's actually not one that you want, and it's one that if you're provided therapy, you might select out a subclone that's basically able to get around that therapy. And that's what these kind of graphs show here on the right, and there's a number of excellent reviews on this. Now, when we sequence this, in the past, we've had abilities such as Sanger sequencing. Those are kind of inherently analog methods, but even when we move to these next-gen technologies in which reads come from a single molecule, we can't tell which cell. And so there's still kind of bulk sequencing. So what can we gain by that? What can we gain? And this view here is kind of an important view because we're going to harp back to it. And it's this view of actually these dots are actually of single cells. And it's a view that has long been had by cytologists and one that becomes relevant. In fact, I'm going to start kind of going back a period of time and looking at one paper that we're going to look a fair amount in, and it has to do with kind of the proof of principle of working with the idea of single cell copy number. And it's a study from 1993 looking at cytogenetic homogeneity of 8 independent sites in a case of melanoma. And in this, George Moore puts forth a series of specimens that over time have become kind of reference standards. And so this is kind of an originating paper for the cell line that we're going to spend a lot of time studying, and a cell line that's been studied by a lot of individuals. And so it's what is colo lines. And we will get into that. But first, let's just take a step back. And I want to take a step back to some earlier studies that we had. These are collaborations when I was at the Translational Genomics Research Institute, TGen, working with the Mayo folks on clinical precision medicine, the idea of applying technologies and making therapeutic decisions on it with Alan Bryce, Mitesh Bora, John Carpton, and a group of us studying a case of cholangiocarcinoma. And in this is one whereby you're looking at copy number traces at this time. And this is basically the idea that we were trying to see where there were amplifications, kind of indicated in red, and where there were deletions of this cancer. We see kind of a spike here on chromosome 8. And that spike is actually, there's a gene underneath it, and the gene is NRG1. So you might interpret this to say, okay, part of chromosome 8 is amplified. The other part might be deleted. It's important to note this y-axis really doesn't tell us copy number. It's actually giving us a fold change as compared to a normal. And so that'll be important later. Now, what really kind of matters from this is that a treatment decision was really made on this case, and the principle was to go after the NRG1. And so following that, there was a series of cytology images taken, and the idea was to kind of look at this not just from this kind of bulk sequencing, but try your best to look at it from a single cell sequencing modality. The treatment occurred, and then we redid the entire analysis. And when we did that, we saw that sure enough, that treatment that was targeting this NRG1 locus was gone, but there was a new peak. What we'd done was subselected a clone. And this time, the idea was to target this clone with erlotinib. And so you can see here, this peak's kind of gone away. We clearly have selected a very different clone. It looks different. And we're able then to kind of verify this by looking at the cytology. And these, again, are single cells. One of the things you're noticing here, and this is going back in time and saying, okay, when we saw this bulk study, we really wanted to get a single cell view to take a picture of what was going on. This is actually part of this study. Now, after that occurred, there was actually another relapse. And so this is a time series in which now we're on the third round, and we've treated 2 different targets. And when it comes back, well, we bring back this locus here. And what also happens is there are some new mutations. And so what you're seeing here is kind of a back and forth with the oncologist looking at the copying of the data and basically giving therapeutic options. And they're effectively doing their job, but there are subclones. And if we can get a better idea of these subclones, we might understand a little bit better how we can treat cancer, especially in very difficult recurrent cases. So once again, we're going to zoom in and see the single-cell traces. So what you see here is this kind of premise where these changes matter. This is, again, evident of a game. We see probably a focal amplification. And these dots here, I think it's just best to take away that they're single-cell validation using fluorescence. So that's a motivating factor, studying and understanding cancer and its heterogeneity. Let's get back to this case that we're going to put a lot of thought into to see and investigate kind of the utility of single-cell sequencing for copy number and DNA. And so this is a cell line that is available through ATCC, Colo 829, and the premise was, if we wanted to, the premise actually for us with this cell line started back with, if we want to take those days of precision medicine forward, we need to come up with a reference standard, something that we could take within clinical labs and validate. And so there was a series of groups and collaborations with the folks in British Columbia, one group, Illumina, and a couple other groups as well. And what we did was create kind of the idea was a reference standard for cancer sequencing. For those of you who might think of like the genome in a bottle, there's this NA1 trait 7A, this individual from Utah that's kind of a reference standard for germline sequencing. That's important for inherited diseases. But actually, cancer, when we're looking at tumor-specific alterations, didn't really have a great reference standard. So this was kind of an agreement between all of us to kind of work together and take a series of samples, basically grow up cells independently, sequence them independently, analyze them in a number of ways, and come up with kind of a set of ones that were reproducible, repeatable. We actually brought in a lot of the original data. This line had been studied before we started this. This was published just a couple of years ago. And it was Aaron Pleasance and colleagues with Mike Senton's group actually went. And so we went and downloaded their data. And what we have here are kind of three growths from a stock, and then sequencing, kind of creating a cross-analysis to identify some true set to publish it. And what we were able to do was really characterize single nucleotide variants and some of the insertions, deletions. We have here the kind of authors. This was actually communicated, Winnie Lang and Sir Nasser, but there's also several other individuals in here. And we would have these meetings and discussion on the copy number. And there were always some things going on that we fully didn't appreciate. And there were differences, too. And differences would be expected. And so we kind of characterized these differences. But the decision was, well, something's different with the copy number. I'm going to show you a little bit of that. Let's just focus on what we understand well, and that were these mutations. So these are some figures that show the differences between these different passages. You're going to see this little chromosome 1p loss here that we put there. It was something that we could noted, and it carried with it a fair number of changes. We kind of expect some differences. But this was one we actually had a fair amount of discussion on at the time, and this is a trace of actually some of the analysis that some of the Illumina folks, Sir Nasser, some of the TGEM folks we would look at, and this was a trace of log fold change versus the normal. In this case, it's a B cell line. And what you see here is, in all of these traces, they look fairly the same. But actually, there are some differences, and the Illumina folks would bring up this fact that there's something going on here at chromosome 1. And there was a lot of discussion of what this is and what this means. It was the TGEN gaining a sample. Was it losing? It kind of looks like it loses an arm here. That was one of the descriptions when I go back and look at the email, is that this part was lost. Now, that was kind of where we were able to leave it. We wanted to get the data out to share it and kind of allow the community to do their own analysis and identify some things. Now, one of the important things that occurred a little bit, not sure about that time, is actually there was spectral karyotyping, kind of the original single-cell method done on this this cell line. And I want to kind of point to this because we looked at this actually after the publication. What we could see is there is a tremendous amount of aneuploidy. You see multiple copies of chromosome 3, 4, 6, 8, 9. That brings forth one of the inherent shortcomings. If we, walk back to this slide, I kept saying that this here, we actually, the y-axis is a little tricky. We can say relative fold change. We actually don't know if it's a copy number gain or not because we have to assume how many overall copies there are because mathematically we just can't derive it. There are various tricks we used, and the BC group, that said we tried 14 different tools, and none of them matched exactly what the true answer was ahead of time. And it's actually interesting. I have a bunch of notes here from Paul Edwards and the individual who put this together, and these are the notes that they had, and they did this great thing, put this forth as is, and they kind of describe a few things in here, and I'm going to highlight a few. There's evidence for monosomal karyotype evolution, endoreduplication. There's a lot of information. There's 311s. You can see that this last sentence here is going to become important. A likely explanation is that the consensus should include a 118 translocation. That's really interesting. So that actually wasn't discussed and thought about when we would look at the bulk sequencing because it wasn't where we were going. It's one of those things which we saw the 1P, but we didn't really see anything going on with 18. It wasn't necessarily the focus. But once we see this, it's starting to make sense. So there have been a lot of papers on this line. I'm going to show you a few, just highlight that they exist. And so there are these efforts to develop these copy number tools that allow us to see the copy number changes underlying a tumor and the heterogeneity. One example here is a CREST paper, which is a algorithm by the WashU folks. And in this algorithm, you can see that they're kind of going through and developing it. They actually do look at colo 829 as kind of their standard. And one of the things that's interesting to look at is you don't really see, you see kind of acknowledgement that there is something else going on, but not an ability to really kind of frame every part of it. There's some other analyses papers. This was actually focused on clonal heterogeneity. This is in Bioinformatics Journal a little bit later. And in this it says, in the tumor sample, we observed that there's events on 2, 3, 7. There's aneuploidy. Assuming that the sample is pure, the speculation is confirmed by a previous study that characterized genome-wide copy number 5, 6. When you read this, you kind of get the sense that it's unclear whether or not there's heterogeneity or not. Something is going on. And so I think that most folks who would look at this in the community, would say that, yeah, there's something going on, but we're just not understanding it. And so this takes us to our method. And that's a lot of background, but you need to understand kind of the insight that kind of comes with looking at single-cell data. And so I have these 3 plots here, and we've kind of talked a little bit about spectral karyotyping. I am by no means an expert in this. In fact, this study has taught me to appreciate how we're in a back-to-the-future moment of appreciating how right this was when we moved to single-cell, because essentially this becomes something that's available for next-gen informatics. And this is something that we can go a lot further than. And we do that with the technology, and what we were using here was kind of one of the very first iterations of the single-cell sequencing of DNA from 10x Genomics. The idea and principle here is to get a low-pass sequencing per genome. The data set that we have is really focusing on sequencing about 1,400 cells, generates a lot of reads, but if you were actually try to figure out the mean coverage per base per cell, it's actually pretty low. Now, we'll talk about that and what that means, but one thing it could mean, if you make a copy number call over several megabases, well, you may be able to add them all up and to identify it. And that's exactly what this second phase, this kind of analysis, focuses on. So I don't go through these sample prep methods. There's better folks to me to kind of explain it. But the key thing is we can get from a cell, and we can actually get to sequence level data where the barcode indicates which cell it came from. And we can put together an alignment of the genome and identify variants. That occurs through an idea of kind of partitioning. I'm going to show you a little bit more on this in a coming slide, in making single-cell copy number calls. And what we want to do is I want to take you to one of these single cells. And so if this was a plot like we were used to, but of a single cell, you can kind of see one big change is the ability to resolve ploidy. By knowing it's a single cell, it's much easier to get an absolute number on ploidy. The y-axis becomes meaningful. That's actually pretty huge because you can say how many copies of the chromosomes are there. Okay, so we can look at if this is showing you 4 profiles, perhaps we can put them all together, cluster them, and identify subclones. And that's exactly what this kind of first graph is showing you, where we've taken all of these traces and we've kind of shown them where this is chromosome 1, 2, 3, 4, and the ploidy is plotted as a heat map, and each one of these traces is just one line there. So we're now seeing a picture that we haven't seen before, but we can clearly see that they're clusters. Now, one aspect we can see that a cluster is, this is showing you post-actually cluster discovery. And that was using an algorithm developed and implemented by the 10x folks, discriminant analysis of principal components. And in this particular plot, we're looking at 2 kind of components, and what you can see is the existence of kind of 4 main clusters. We could do further analysis and shows that there's an optimal number, but we could see that some of the cells are falling here, there's a group falling here, and a group falling here, and a group falling here. That's pretty neat. So what that's saying is that our groups are different based on chromosome 1, and actually a little bit more complex than perhaps even we were thinking about. So let's go ahead and dig a little bit more into this. So now we have 4 major clones. And when I saw this plot, it was like, aha. And that's actually where I kind of started working with this data was we had sequenced it, we looked at this, and they were like, what do you make of this? I'm like, I knew there was heterogeneity, and wow, I can see things I never saw before. So that brings us through this analysis of what we wanted to do, and we worked with textbooks on, was clustering these, taking these BAM clusters and creating, at that time, little mini BAMs. And the idea we had there was, let's create a BAM of cluster A, B, C, and D, and look at variants. First type, obviously, we'd look at were single nucleotide variants. We'll discuss as we go, but also copy number variants, looking at anomalous read pairs that map to different places, structural breakpoints. The idea then is some of these BAMs would contain a bunch of cells. Some would contain a fewer cells because it happened to be for the smaller group. So we had 4 BAMs. And those 4 BAMs were the 4 major groups. And we basically treated this like we would anything else and basically do analysis of one versus another, one versus the control, various scenarios. So this then allowed us to look at something important, clones. We've now discovered some clones. Can we gain any insight that we didn't have before? And so here you're seeing a picture of some of these clones. And I like this picture a lot. This is actually a picture in chromosome 18 because you can see so many features. So this is one of our clones. And what you can see here is this is probably over a 10 kb region. We could see a stacking of reads. And the stacking of reads actually suggests a deletion. We've got coloring such that we can see anomalous reads, reads that go from here to here, the red reads joining each other. See these guys right here. This is a polymorphic SNP, an inherited SNP right there. You can see how it would be heterozygous, but you can kind of see here, and if you map a whole bunch of these, you would see that there's a loss of the heterozygosity. It's basically looks like this clone only has really one of the 2 parental ploids. This one here, it's harder to see, but if we looked at enough of them, we could stack them together, look at a loss of heterozygosity analysis. This, and this. So one of the things that's really nice is you get a sense of the accuracy of the clustering, by the way, because if the clustering did not use a lot of this information, what you could see is in this 10 kb region, and the clustering that's based on a much bigger window, we don't see random reads floating in there. That's one reason why I was quite excited about it, because it looked like the clustering was kind of interesting. But let's look at actually this key region, this jump from chromosome 1 And one of the things we did was we tried to zoom in here, and what we were able to observe was a translocation to chromosome 10, 10p11.2. And so now this is a new picture, and we've zoomed out. This is actually then a kind of the break on these different clusters. And so you can see some of them, you see the drop, and some of them you don't. That's really exciting. We can zoom in further. We can get base level resolution. And when we zoom in far enough, if you're used to these traces, you're probably wondering about those. These are what's usually thrown out. So this is a strictly BWA alignment, but these are basically then the reads where every single base doesn't match. That's the point, the exact point where the translocation occurs. And so we're able to get single cell base level resolution of these. Now each one of these reads may come from a different cell. Remember, we're looking at a collection of cells, but we still have that ability. So what you can see here then is the ability to then map translocations down to a single molecule level. And what we're depending there on is by having enough really kind of cells in the group to identify them. We are able to go a little bit further and find another translocation that jumped from 10p14 to 18p11.32. This is where the real insight came in that we never had before. What this suggested that we had was a hybrid chromosome from chromosome 1 to chromosome 10 jumping to chromosome 18. So here, here, here. This actually flips around. It's actually a kind of derivative of chromosome 18. And this is something that Paul Edwards kind of talked to us about when we kind of went to him and talked to him about his results. So we have this picture of seeing all of this. Now let's go back to our original data. I said we saw this stuff before. And so we went back to the bulk data from back in the day. This is 2012 actually, even though the paper was published later. What we can see is that the events that we're seeing here were found in the previous sequencing, and that the TGen line didn't show the hybrid. So what it suggests is that this hybrid was somehow lost. And that kind of makes sense because this would be largely unstable and easily lost. And this actually brings us to this overall model, this picture of the chromosome plots, where we can then understand and build things up. And I'm going to fold in one last piece of data. It's really rich data, so you can fold in a lot. Remember I talked to you about loss of heterozygosity? This plot here, we start looking at a mulch. We have single cells. This is actually the trace. This is developed by Jessica Aldrich, which was a tool kind of looking at copy number, where you can see these events, but we've actually mapped what we're calling allele frequency or loss of heterozygosity. And what you don't see are them tons and tons that actually jump to 0 or 1. That's why we put these numbers here. And we also see something on chromosome 8. And so we can identify differences between group A and group C. We can identify differences from SNPs, which is loss of heterozygosity. We can find structural breakpoint differences, and we can find copy number differences. When we look at the bulk sequencing, we can do something similar. So just to make sure I've covered every part of this figure, this is how many reads are jumping between the two chromosomes that we actually see the mapping for. There was a part where we see an additional event occurring on chromosome 8, and this event's also lost. And there's actually a really elegant explanation for a lot of this. But one thing I want you to notice, some of the parallels are that effectively at the same points, and they are actually at the exact same point, just like the rendering, we see that the TGen bulk actually is very similar to one of our subgroups, this group C. And so it's fascinating to see it recapitulated time and again, but it showed kind of a general model that was overly occurring again and again in this model where we have kind of a starting cell and we're growing it. It starts out with this 1p10q 18q, this aberrant chromosome 18 that's often lost. And so several of the sites which had the bulk sequencing had all of them, but some of the other ones didn't. And so that gave us an insight we didn't have before. And what I like about this is it shows the richness in a very boring topic, it's cell line growth. So Now I'm going to jump into some things reason why we were doing this in the first place, which was to move to patient samples. So the idea that we use this colloid 29 line, we were able to get insights and explain things we weren't previously able to. We took, in this case, and this is actually a little bit a while ago, a set of renal cell carcinoma dissociated cells and had them sequenced as well. And with this single cell method, you can see the number of cells that are being sequenced here. It's a fair bit lower because, when we thought about it, we really we didn't need that many. One thing you could do, just like with next-gen sequencing, is kind of tune this towards a goal. But the sequence in the neighborhood of 750. I'm going to show you each one of these. And, there'll be this part where I think some individuals will immediately say, well, I see what's going on in there. And one thing is, if we're looking at a regular specimen that came from a patient, we might see a mixture of the stromal cells and the tumor cells, a tumor normal mixture. And that's actually what you're seeing here. And so if you look at these are basically traces, and we could say this is about half is diploid and half is tumor. And we do see actually some evidence for kind of clusters, right? We see these kind of blocks. This one, we actually see only a little bit of tumor, about 15% of the cells. Here, we see about 20% of the cells. What's really great about this is there's a lot of studies which talk about tumor-only sequencing, and here we could just focus in on these tumor samples. We could identify those mutations that were specific to the tumor. Exactly what we did, by the way. I want to dig in there and show you that. First thing, though, that was obvious to us was that we were able to see distinct chromosome 3 profiles among all of the different cell groups. And what we could see is on these different cells, we could see this kind of core event for us, which was on chromosome 3, which contains this kind of key driver mutation in VHL1. Now, one question you might have is, could you detect a variant? Could you detect it in a small portion of your cells? So what we did was, in this case, we ran the stroke. We created a little mini-BAM of this slice of 51 cells versus all of these 800, and then looked at the calls. And so what did we identify? One of the variants called, actually with a fairly low number of reads, was one of the canonical driver mutations for this. And so it's kind of neat to be able to see this actually being called. Now, there's a stochastic nature of that by the fact that we're looking at something that's probably 10% of cells, but we're able to pick it up, and actually it's a heterozygote. So this is actually, we were able to show that as well. This is another case where we're actually making an indel being called from the non-diploid and diploid. These are inverted, and you can see that there. And so now in this case, it's a little bit more straightforward. So that actually shows you something that you kind of was hoping to see. We didn't see it with colloid 29 because during a cell line growth, you probably don't acquire a ton of mutations, and we kind of didn't expect to. It's really, though, the tumor evolution that's occurring within a tissue that we wanted to see, and in fact, we can see. So that kind of brings us to the end of this talk, and really, there are some conclusions. This talk could be called Back to the Future at some point, but single-cell sequencing of DNA significantly improves our ability to understand tumor evolution and tumor heterogeneity, particularly those tumors that are driven by aneuploidy. And that identification of these CNA clones, our groups, really opens up subclonal analysis of structural variants, SNP through LOH mutation. The SNP LOH can identify and track chromosome loss and gains. I feel there's a lot that can be done on the analysis side, for which I'll never be able to pursue, but I'm hopeful that somebody will. We were able to use breakpoint analyses to show things that we weren't able to show before and distinctly link two regions, something we could not necessarily, we didn't do before, and get a clearer picture of tumor clonal evolution. And the one thing it does is it helps you understand what is really going on here. So this brings me to my last slide, and I want to thank you guys all for listening with me. This is a lot of the work is led by Enrique, who worked closely with the 10X team, and really the key person that was interfacing with us and implementing and developing a lot, particularly in the classroom, was Shamini, pictured here, but with a team of folks, including CLIA on the experimental side, DJ on the computational side, and probably a bunch of other people that aren't here. Some of the key insights also came from Paul Edwards, who really was just wonderful to put out so much of his data online such that we found it and gained insight. I think that was really an incredible part to that, and it was really through Mira's work. The single cell patient data we saw was Lee and John's, and then we also brought in Jessica to tweak the LOH algorithms a little bit to get those to work and bring that in as an addendum. And so we have all of this, the paper is in review, but of course we do put things out in pre-publication, and that'll be available as well. So thank you for your time.
Thank you, Dr. Craig, for your presentation. We will now start the live Q&A portion of the webinar. A quick reminder for our audience on how to submit questions. Simply type them into the drop-down box located on the far left of the presentation window labeled Ask a Question, and click the send button. Dr. Craig will answer as many questions as time permits. All right, let's get started. Our first question is, moving beyond cell lines, what other applications are there for an assay like this?
So thank you for that question, and that's really an important question because it's at the crux of why I'm enthusiastic about the ability to look at single cells, and really the applications are in understanding tumor evolution and tumor clonality within specimens. Most of the time when we're looking at a specimen that's been taken from a patient, it's oftentimes not just pure tumor. There's often normal stromal cells, as we saw, and there's also going to be a tumor evolution that's occurring, and we will see cells from all of those. So what we're excited and what we've been working on the most is really implementing this in the context of different tumor specimens, and that's really where the excitement is at that we're currently on. So we're looking at endometrial, we're looking at ovarian, prostate, breast cancer, and to try to understand within a course of a tumor, particularly those that have been treated multiple times, do we see multiple clones, and can we resolve them using this copy number driven approach to resolving the heterogeneity.
Great, thank you. Okay, our next question is a two-part question. What are the limitations of bulk-seq for this, and can this same work be done with tumor normal samples and informatics methods?
So that's a question that gets to the heart of, the idea that if we push a bunch of things together, what can we resolve in terms of a mixture? And one part of that is whenever you see this presentation or any others, it's always like, okay, well, we can see that in the bulk, but could we have discovered it? And that often is not the case. And in this particular example, this tumor had been sequenced multiple times by multiple groups at very deep. And we clearly see evidence for all three of it, but we were never able to really resolve all the features that were going on. And so there's certain things where you just lose information. And so in large part, understanding clonality informatically is not something done routinely. So in other words, informatics really hasn't gotten to that point of being able to do it perfectly. There is some really work we have published on this. This work was led by Rebecca Halperin, who is a faculty member out of TGen, where she looks at how well we can do. In that, we found that we had to go and sequence very deep, around, 800x, and we would have to model the copy number in the SNPs. But and so depending on the nature of it, we were able to see some, but not all. That in itself means that we're kind of spending money elsewhere. So there's a certain big advantage to this. So at a certain point, we can definitely do this and resolve some aspects informatically. but it's nothing that compares towards the ability once you already have these separated. So if you do want to see kind of the extent of that, you can look up Halperin's paper on this that we wrote together on it. And you can also go through the positive predictive value in all the places where things don't work out well, where they do. But there's a reason why, having developed those methods, that we are enthusiastic about being able to directly do the single cell sequencing of the DNA, and it really does aid and actually show insights that we never saw before, as evidenced by this particular study of kind of our benchmark, right? So there's just certain things we cannot get.
Great, thank you. Okay, your next question, how many cells can I load into a chip channel, and what recovery rate should I expect?
So there's a certain part I'm going to defer to some others I've asked to be on the phone call, but the broader question gets into how many cells do we go after. And we've taken different approaches to this question. And in some cases, what we've done is we've set things up so that we could do more cells or fewer cells. And a lot of it depends on the nature of the tumor that we're studying, what our goals are. In other words, what sort of subclones to pick up at. And for many of our studies, we've kind of designed for 500 to 1,000. For some particular ones, we do want to go a little bit more. It's very much dependent on the context of the assay. Now, I want to take this part, and because there's a technical aspect to it, I will defer to colleagues I've also asked to be on the phone from 10X, and so maybe they can pick up from there on the technical details.
So for our CNV assay, we recommend loading from 1,600 as a minimum up to 31,000 cells as input, and you can expect up to a 15% recovery rate after sequencing of those inputted cells. We do have a technical note new on the support site for the CNV assay section, and it will help you to select number of cells if you have other data as far as if you know what size events you're looking for, and also sequencing considerations.
And then to add to that, when I said the 500 cells, usually that's what I end up looking for at the target. So actually, we would be loading in more. It's one of those, when you get the PI answering the question, all you often care about are the results, how many end up on there. And so I think that's exactly why I wanted the 10X folks to jump in on that one to help out with those details.
Thank you. All right, next question. If I decide to reduce sequencing, is that okay? And what will be the trade-off?
So, I think with a lot of next-generation sequencing, one of the key aspects is that fundamentally it was a shift going to sequencing kind of these quantum or these singular events, and that allowed us for the first time to be able to really control error towards our goals. And what I mean by that is, if we think about some of the original, the projects like 1,000 genomes, it was 4x sequencing, which seems kind of crazy, but to identify all of the polymorphisms within a population greater than 1%, really that was all that was needed. So the original project there was 4x. Now, when we talk about sequencing individual genomes, there's a recommendation that's 100x. That will get you fairly good accuracy. Now, in a clinical lab, often we do exome sequencing that gets to 80x, 90x, 100x, for example, and that really gets you to another level of quality. And so these same parallels, I think, have the potential to exist with single-cell sequencing. When I think about this, and I think about our specific utility, we're often going to be looking at kind of solid tumors and looking at heterogeneity and trying to identify clones, subclones that are in the neighborhood of about 5 to 10%. So we can kind of model that into how many cells we think we would need to be able to get, and in the end, that determines a lot of our kind of costs and so forth. I think so that kind of frames it in terms of how many cells we want to look at. And the example would be if we really want to identify, for example, a mutation specific to a clone at 10%, I'd like to really see, a good number of reads there. And so I might be wanting to see 150 cells or 200 cells or something like that to be able to identify that, which then would mean that, well, probably going to need upwards of 12, 14, 1,500 cells, right? So it kind of really depends on the goals. That's in the case of trying to identify a single nucleotide variant. So there's some variability, and the way our experiments are designed, we often retain the ability to go back and either experimentally through the sequencing or going back to the assay, go deeper into that specimen in a second round. So oftentimes we do kind of to the ability to take certain samples and look at them at a deeper coverage by adding additional experimental data later. So that's kind of how we attack it, by basically having a baseline, and then if we need to, for a specific study, go deeper through additional experiments. And that's one of the effective ways we use to manage kind of the overall costs. I don't know if 10x has any thoughts on this in terms of their recommendations. That's kind of a specific kind of example in our case. Anybody from the 10x have more broader answers towards optimal study design?
We would recommend customers refer to the tech note that I referred to prior, just depending on what information they have and how they can frame their experiment. That document's really useful, which touches upon sequencing, number of cells to be inputted, as well as what event size you're looking for, if that's on a single cell level or if you're okay with looking across a cluster of cells.
I think one of the things that's, kind of everybody has something different they say. And so for us, you'll see, at least some of my thoughts, you'll see a lot of bias towards seeing kind of collections of cells. In many of our studies, if we will only see an event in a single cell, I don't think that we can experimentally interpret it, or at least we're not planning to. It's really when we see kind of the suggestion of a subclone that's, somewhere in the range of 1 to 5% that we'd want to look at. Now, one could see and speculate and find scenarios where a single cell is informative. It's just not part of our particular experimental design.
Great, thank you. All right, your next question. What is the largest cell size I can use for this assay?
I have no idea, and I would really like to hear the 10x folks answer that. I have not thought of that question.
So we've tested up to 30 micron diameter size cells with our CNV solution. If you're testing something larger than that, we generally recommend isolating nuclei. And we do have a protocol on the support site that addresses frozen tissue as well as fresh or cryopreserved cell lines. To give you an idea or an outline of how you can go about prepping cells or nuclei from your tissue or your cell line. If you do use cells that are larger than 30 microns, you risk clogging the channel of the chip, and therefore you'll get a clog and you won't be able to recover your emulsion and proceed with the assay.
Thank you. Okay, can I aggregate multiple libraries that were sequenced from different parts of the tumor to create one set?
So this is a really interesting question, and I think one of the core aspects is the heterogeneity, particularly in solid tumors. And there's a lot to that, and I think different fields of different expertise, they may be talking about, kind of at the edge of a tumor, at the boundaries, that maybe there are different cells, and there are all these interesting questions that have to go in terms of overall tumor metastases. And so for a large part of many studies, you can imagine scenarios, and there's a great work by Christina Curtis that kind of is published in Nature Genetics, talks about this Big Bang theory of of cancer genomics, but you can also see it seated in so many others works where you're going to see underlying heterogeneity, and it may be different in different parts. And so it would be really great to look at different parts of the tumor. And what you would be curious about are, would some of the dominant clones be present in, let's say the boundary, the leading edge, versus the kind of, let's say, bulk of the tumor? That's kind of interesting. There's so many different aspects that we think about with this, and as far as how do we bring them together and aggregate them, well, I think that we haven't explored all the different ways we can do that, but I can definitely say that being able to look at them all together, I would suppose that I might try to actually aggregate them all and do the clustering together, and I would try to not have it together. It's a great question. It would be part of a strategy. Analysis-wise, I don't know if anybody on the 10X side who did the clustering would have any opinions if they took two different parts of the tumor. I actually might try to have them aggregated. We'd try to do both. Anybody over there have any thoughts on if you would sequence like the leading edge of a tumor in the middle of the tumor, whether or not you would run it as a single analysis or kind of keep them separate and then compare?
Yes. Hi, this is Andrew with 10X. I'm in the bioinformatics support group. Yeah, we actually earlier this year released a new pipeline within Cell Ranger DNA called the Agri pipeline, and this allows the user to aggregate multiple libraries together into a single set of output files up to 20,000 cells. And we actually have a new tutorial available online now that illustrates how to do this using a breast cancer data set where we have multiple sections from the same tumor that have been aggregated. So you can find that on the 10X Genomics support site, clicking on the single cell CNV solution, software, and tutorials.
So, Andrew, I know we have a little bit of time. Can you tell us then whether or not you think that you would be able to resolve things that you wouldn't normally by putting them all together?
Well, at least in the breast cancer data set that we looked at, we can certainly see that there's like an additional level of heterogeneity that wouldn't be obvious from just analyzing any single library. So yeah, there is variation like spatially across the different sections of the tumor.
Okay.
Great, thank you. Okay, it looks like we have time for one more question. And this question is it possible to get SNP information from this data for mutation detection?
So that question has a little bit of complexity to it. So a SNP, single nucleotide polymorphism, is typically something that's polymorphic and inherited throughout a population, and so that would be a germline event. So in our presentation, we do go and we look at where these go from being heterozygous to homozygous, like from the deletion. But I suppose the question is probably more actually looking at single nucleotide variation or insertions and deletions are specific to a clone, a mutation driving a clone, for example. And so that's something that within the CLIA-29 we did not observe, and you might think, okay, that's reasonable considering, for the most part, you were looking at comparing different clones and you're only looking at 10 passages. But actually, very much so, we can, in various studies, we have been able to identify mutations specific to a clone. I know that we've been using kind of our own in-house approaches to these, but largely that have aggregated together BAMs and then being able to do mutation-specific calling, comparing them. I know that we had conversations with the 10X folks about them kind of creating additional solutions to doing mutation-specific calling. But let me tell you, that's one of the reasons why this is so cool. I mean, if it was just CMVs, then that would be good. But what's really great is to be able to use that and the subclones to identify those mutations. And so, yes, Yeah, very enthusiastic yes to that. It depends on number of cells, of course. And then I'm curious on the 10X side if they have suggested any like supported solutions.
Hi, yeah, this is Andrew again. So we heard during the webinar from Dr. Craig about how his team analyzed SNVs for subclones. So just wanted to point out that in the latest version of Cell Ranger DNA, we offer a BAM slice pipeline, which was not available at the time when the paper was done, but it basically allows us to subset the BAM file, which contains all reads in the data set, by the cell barcode or the 10X barcode. And this way, the user can do their own SNV calling on the subclones using third-party tools. And we do have, actually, Dr. Craig presented this earlier in the webinar on the Colo 829 data set, splitting up the BAM file by the 4 groups A, B, C, and D that were identified with discriminant analysis of principal components. So in that tutorial I just mentioned in the last question, we also have an example of how to use the BAM slice pipeline to subset that BAM file by the 4 groups that Dr. Craig mentioned specifically. And we also noticed that, another preprint came out yesterday from Dr. Ben Raphael's group using a novel method called Chisel that uses low coverage single-cell genome information to call SNVs and determine clonality. So that's also really exciting, something I haven't looked into in detail yet, but as it just came out yesterday. But so this field is evolving quickly.
Yeah, so the one thing I would add to what Andrew just mentioned, I haven't looked at Ben's preprint, but for the SNV calling, I think we didn't mention it, but we've looked at a few different tools. Pragmatically, we have had the best success just using Straka, which is a tool that's implemented by members of the Illumina informatics team, and we found that that works very well without any need for additional filtering. We also looked at Mutech. That worked as well. Required a little bit more filtering. I kind of was enthusiastic about the Straka just because it does some kind of micro-alignment or microassembly within it. So I think that there will be multiple approaches to the mutation calling. I could just say that, you know, pragmatically, we have been having success just breaking out the Straka tool and applying it straight up to the slice BAMs. So that's kind of been how we've been attacking it. It seems to be working very well.
Thank you. I would like to once again thank Dr. Craig and the 10X Genomics team for their time today. Do you have any final comments for our audience before we go?
I don't. I think as we enter this kind of new era of moving from single bulk collection of cells to single cells, we're seeing kind of the same sort of revolutionary ability to see things that we couldn't see before that we saw when we moved into next gen, you know. So next gen sequencing was about sequencing of molecules and looking at the underlying molecular events from a singular point of view, and now we can do this at a cellular level, and now really it's the DNA that's allowing and starting to drive these as well. And so it's taking a step beyond single-cell RNA sequencing, and there's quite a bit that can be understood from sequencing of the DNA of single cells. Even when it often technically has challenges, we're able to resolve very important disease-relevant problems that have been core to our field, particularly cancer genetics, for so long. So it's exciting kind of early days of that. And I think that there's a lot of great stuff to come.
From the 10X support side, I would just say if you're planning experiments and you think the CNV solution is something that maybe can help you answer your questions, please email us at support@10xgenomics.com, and we'd be more than happy to help you work through that experimental design or help you answer specific questions about the workflow or the bioinformatics end. Thank you.
Thanks so much. I'd also like to thank the audience for joining us today and for their questions. Any questions we did not have time for today will be addressed by the speaker via the contact information you provided at the time of registration. We would like to thank LabRoots and our sponsor 10x Genomics for underwriting today's educational webcast. This webcast can be viewed on demand, and LabRoots will alert you when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's event. Until next time, goodbye.